Okay. So, when a sperm is coming, when a sperm is coming and the sperm is reaching near the ova, suppose it is a ova, okay? By covering the ova, you know very well about the secondary envelope. That is a secondary envelope is there. The secondary envelope in the case of this ova is known as egg jelly, right? There is egg jelly as the secondary envelope. The egg jelly contains certain polysaccharides. The egg jelly contains certain polysaccharides. These polysaccharides are present in egg jelly. These polysaccharides are known as sulfate-containing polysaccharides. Egg jelly contains some polysaccharides that is known as sulfate-containing polysaccharides. So the sulfate-containing polysaccharides are present in the egg jelly. The egg jelly will be containing sulfate-containing polysaccharides. So the receptors for this, the receptors for the sulfate-containing polysaccharide that will be present here on the acrosome. On the acrosome, here it is the acrosome. Just above the acrosome, you can find the receptors. You can find the receptors for whom? Receptors for the sulfate-containing polysaccharide. Sulfate-containing polysaccharide will be present on egg jelly, and their receptors will be present on the sperm. So, whenever they come in contact with, the receptor will attach with the receptor will come and they bind with the sulfate containing polysaccharide. So, there will occur a binding. A binding will occur there. Due to that binding, what will happen, Due to that binding, three changes will happen. The first one is that activate a calcium channel. That activates or open due to that binding that opens a calcium channel. One calcium channel will be opened. That will open one calcium channel. Due to the opening, due to the opening of this, what will happen, due to the opening of this, calcium enters into sperm head. Due to this, the calcium enters into the sperm head. Into the sperm head, calcium will enter. Whenever the calcium, what is the role of calcium? Calcium is needed for vesicle fusion. For vesicle fusion, anywhere in our body, for vesicle fusion, calcium is required. So what will happen, whenever the calcium enters into the sperm head, whenever the calcium enters into the sperm head, what will happen, the acrosomal membrane, this is acrosomal membrane. This acrosomal membrane will fuse with the acrosomal membrane. Acrosomal membrane, what will happen to the acrosomal membrane, acrosomal membrane fuse with the plasma membrane. Plasma membrane of the sperm. Acrosomal membrane will fuse with the plasma membrane of the sperm. So what will happen, this acrosome, listen here, this acrosome will be fusing with this acrosome, this is acrosome. This acrosome, what will happen to the acrosome? That will fuse with the plasma membrane. This was acrosome. This acrosome will fuse with the plasma membrane. The acrosome will fuse with the plasma membrane. That will happen. Then what will happen, after this, what will happen, the acrosome slowly extend outwards. This acrosome, this acrosome will be slowly extended. Now it is facing inward, right? Now it is facing inward. From this point, they will protrude outside. They will protrude outside, and in this manner, they will be protruding outside. They will form a structure in this manner. So acrosome extend outwards. This acrosome, this acrosome that extend, the acrosome extend outwards. Acrosome will extend outwards. What will happen inside this, for that, inside this, actin polymerization will occur. Due to actin polymerization will occur here. Actin polymerization. Due to actin polymerization, the acrosome will be extending outwards. Acrosome will extend outwards. So, in acrosomal process, in this acrosomal process, there will be another chemical substance present here. Another type of chemical substance will be present here now. It is known as bindin. It is known as bindin. Bindin will be present here. Bindin will be present here. So the binding will be present here, that is exposed. So the binding which was present inside now exposed outside, right? It was exposed outside. So what will happen, now a particular structure is formed here, a particular structure is formed here. That structure, the acrosomal process, that is the acrosomal process. The acrosomal process, what they will do, the acrosomal process cleaves, they will cleave the egg jelly. Acrosomal process will cleave the egg jelly. Egg jelly is present there as a secondary membrane, right? They will cleave the egg jelly and expose, the primary envelope, and they will expose the primary envelope. Primary envelope will be exposed. What is a primary envelope? That you know very well. It is vitelline membrane. So now the vitelline membrane will be exposed. So what is the role of the binding protein now? A binding protein is there, right? The binding is required for, binding is needed for, the role of binding is that binding is needed for sperm binding. Binding is needed for sperm binding to where, sperm binding to vitelline. For sperm binding to vitelline, binding is required. Once again, we can look. Sperm is coming. Here it is a sperm. Sperm contains certain receptors. And the secondary envelope of the ova, that contains some polysaccharide, that is known as sulfate containing polysaccharides. Whenever the receptor attaches with the sulfate containing polysaccharide, what will happen? One calcium channel will open there. Whenever that calcium channel open, what will happen? Whenever that calcium channel open, calcium enters into the sperm head. Whenever the calcium enters into the sperm head, what will happen? The acrosome will fuse with the plasma membrane. Acrosome fusing with the plasma membrane and acrosome, due to actin polymerization, acrosome will form a projection outwards. The acrosome is extending outwards now. Now a protein which was present inside is now exposed outside. That protein is known as binding. The name of that protein is known as binding. What is the role of binding? Binding is required for binding the sperm into vitelline membrane. Sperm want to bind into the vitelline membrane. For that, the binding is required. So up to this, the primary envelope. So by using this structure, they will cleave the egg jelly. And after cleaving the egg jelly, the binding will help the sperm to bind to the vitelline membrane of the ova. Whether it is clear?
Yes, sir.
Okay. Okay, ready. So listen, for binding, binding is present in a sperm, right? For binding, there is a receptor in ova. There is EBRI receptor. That is a EBRI receptor. This EBRI receptor, where this EBRI receptor will be present? This EBRI receptor will be present in? This EBRI receptor will be present in? Tell me. Binding is attaching to EBRI receptor. So EBRI receptor will be present in? Ovum or vitelline membrane of the ovum. EBRI receptor will be present there, isn't it? EBRI receptor will be present there. So the binding will be coming in binding will come and bind with the EBRI receptor. When it is coming in bind with the EBRI receptor, what will happen, they will form inside the ova. In ova also polymerization happens. In ova, a polymerization happens. Polymerization of actin. In ova, polymerization of actin and actin will polymerize and form a microvilli-like structure. A microvilli-like structure. The ova will form a microvilli-like structure. So suppose it is a ova, okay? Here it is a ova. After the binding, from this portion, they will form a microvilli-like structure through actin polymerization. They will form also a microvilli-like structure they will form. So the sperm is interacting with the sperm is already having a particular structure in this manner, right? Sperm is already having a particular structure in this manner. Acrosome was protruded outside. So the sperm is interacting with this microvilli-like structure where there is a fertilization occurs. So this region, this portion we are calling it as fertilization cone. This structure is known as the fertilization cone. This cone can be called as fertilization cone. This is known as the fertilization cone. So in fertilization cone only, who will be attaching sperm and ova will be coming and attaching in the fertilization cone only. So that region that is known as now as fertilization cone, that is fertilization cone. That is also clear. In fertilization cone, what will happen, in this fertilization cone, in this particular region, a protein will be there. That protein is known as fusogenic protein. That protein is known as fusogenic protein. What is the role of the fusogenic protein? Fusogenic protein, they will fuse. Fusogenic protein will fuse the membrane. Both the membrane, who will fuse the membrane, fusogenic protein will fuse the membrane. As a result of the fusion, here it is a sperm head, and here it is that fertilization envelope, fertilization cone, and here it is a ova, and here it is that fertilization cone. They will fuse together. They will be fusing together. They are fused here. Here it is a nucleus present here, and here it is a nucleus present here. So what will happen, this is a egg or it is a ova. So after the fusion, now the path is very much clear. So through this path, who can move through this path, the male nucleus can move into the female, move into the egg. The nucleus from the sperm that can now easily move towards the egg. Whether it is clear?
Yes, sir.
Okay. Whenever this happen, whenever this process happen, the egg will prevent. Whenever this happen, egg will prevent the entry of further sperm. Only one sperm can fertilize. An ova. The egg will prevent the entry of further sperm. So we are having the monospermy. Monospermy means only one sperm will be binding with the ova. Monospermy. How the polyspermy is prevented, Polyspermy, what is polyspermy? Multiple sperm is coming to an ovum. Polyspermy in sea urchin. Is prevented by, how it is prevented, is prevented by two mechanisms. There is two mechanisms. By these two mechanisms, the polyspermy can be prevented. Polyspermy can be prevented by two mechanisms. What are those mechanisms? We can discuss now. How the polyspermy is getting prevented. Listen to me. Polyspermy, for preventing polyspermy, the first method is known as fast block. There is a method known as fast block. That is a first method, or it is also known as electrical block. Fast block or electrical block is a first method to prevent polyspermy. We know very well that a normal membrane potential, a membrane potential, a membrane will be always in a polarized condition, that you know very well. The normal membrane potential is 70 minus 70 microvolt. When a sperm bind, when a sperm bind is binding, when a sperm bind, they will activate when a sperm bind, they activate a sodium channel. When a sperm bind, activate Na plus channel. Na plus channel will be activated. Whenever the Na plus channel is getting activated, whenever the Na plus channel activated, normally it was in minus 70 microvolt. Whenever the Na plus channel is getting activated, what will happen, Na plus will enter inside the cell. Na plus will enter into the egg. Na plus will enter into the egg. So what will happen, the membrane potential will become now, due to the entry of the Na plus, the membrane potential will become as 20 microvolt. It will become as plus 20 microvolt. So, whenever the sperm and ovum is coming, actually, whenever the sperm is coming, sperm is also having, sperm membrane is also having minus 70 microvolt. This minus 70 microvolt can fuse with this minus 70 microvolt. But after their entry, as due to the presence of the sodium plus channel, Na plus channel, what will happen? Na plus enters inside and the membrane potential will become as plus 20 microvolt. Whenever it becomes as plus 20 microvolt, no other sperms can enter. No other sperms can enter. Whether it is clear, it is known as the fast block. This is a first method to prevent polyspermy. To prevent polyspermy, there is a first method by changing the membrane potential. Clear? Hello, clear?
Sir, once more, sir.
Okay. So this is a sperm, okay? This is a sperm. This is a sperm. The sperm membrane potential is minus 70 microvolt. This is ova. Ova's membrane potential is also minus 70 microvolt. So the first coming sperm, as the membrane potential of these two, membrane potential of sperm and membrane potential of ova is same, this sperm can easily come and bind here. So whenever the sperm bind here, whenever the first sperm touches here, it will activate sodium channel. So Na plus will enters inside. As a positive charge enters inside, the ova will become as plus 20. Ova's charge will become as plus 20 microvolt. So the coming sperm, the further sperm will be coming with minus 70, right? Further sperms, all the other sperms are coming with minus 70 only. So the minus 70 cannot join with this plus 20. Only minus 70 and minus 70 can join. Only the same charge can join. Different charges cannot join. So it will prevent the entry of sperm. Whether it is clear?
Yes, sir.
Okay. So another method. So this can happen, this can happen in the case of, marine condition. This can happen in the case of marine condition when there is maximum Na plus is available. What will happen in the case of, freshwater? What is happening in the case of freshwater organism? What is happening in the case of freshwater organism? So the sperm, here it is the ovum, a sperm is coming. What will happen, activate in freshwater, it activate Cl minus channel. Chlorine channel will be activated. As a result, Cl will go outside, chlorine will go outside. Whenever the chlorine goes outside, chlorine is negative charge, right? Whenever the chlorine goes outside, as chlorine is negative charge, when the negative charge is moving, the membrane will become as plus 20. Membrane will become as plus 20 microvolt. As it is becoming plus 20, further sperms cannot enter. Additional sperms cannot enter. Sperms always coming with minus 70 microvolt. They cannot join here. This is a fast block. This process is known as fast block. This is known as fast block. This will happen. The fast block will happen 1 to 2 seconds. 1 to 2 seconds after. Fast block will happen 1 to 2 seconds after sperm entry. After the entry of sperm, within 1 to 2 seconds, the fast block will happen. This is a fast block, immediate block. One more blocking mechanism is there. The second blocking mechanism is known as slow block. That is slow block. Slow block is also there. Slow block is another ways it is known as mechanical block. Slow block or mechanical block. Slow block or mechanical block, or it is also known as permanent block. Permanent block is there. What is a slow block or mechanical block or permanent block? In that there occurs cortical granule reaction. Cortical granule reaction is needed for this. Cortical granule reaction is needed for the slow block. Cortical granule reaction is needed. You know very well what is cortical granule. There is in sperm, there is peripheral cytoplasm and all those things we have already discussed about. Okay. So whenever the sperm enters, whenever the sperm comes into the ova, sperm is coming into the ova. What happens, calcium channel opens. Calcium channel will be opening. As a calcium channel opens, the cortical granule that is present in this region The cortical granule, this is known as cortical granule, as we have already discussed about this. This is cortical granule. This is cortical granule. What happens, the cortical granule will fuse. Cortical granule will fuse. So if here the sperm is entered here, the cortical granule will fuse with the plasma membrane. Cortical granule will fuse with they fuse with what happened? They fuse with the plasma membrane. Cortical granule will fuse with the plasma membrane. They will fuse with the cortical granules fuse with the plasma membrane. With the plasma membrane, the cortical granule will fuse. Whenever the cortical granule fuse, what will happen, inside the cortical granule contains a cortical granule. If you are looking into a cortical granule, the cortical granule contains the first chemical that is known as cortical granule. Cortical granule, the first chemical is cortical granule serine protease. Cortical granule serine protease is present inside the cortical granule. What they will do, the cortical granule, what they will do, they will cleave, the cortical granule serine protease will cleave the post or the connection between, they will cleave the connection between plasma membrane of the ova and vitelline membrane. Plasma membrane of the ova and vitelline membrane. Plasma membrane, you know very well, that is covering the ova. It is a plasma membrane. By touching it, there were what? By touching it, close proximity, they were what? By close proximity, there were vitelline membrane also. So this is plasma membrane, and there were outer the plasma membrane, there is vitelline membrane. What they are doing, the cortical granule serine protease, they will separate the plasma membrane and vitelline membrane. So vitelline membrane will be separated from the plasma membrane. That is the first thing they can do. What they will do, another thing that contains mucopolysaccharides also. This cortical granule is fusing here, that contains mucopolysaccharides. The another thing that contains mucopolysaccharides. Mucopolysaccharides are also present there. Mucopolysaccharides are also coming out. What is the role of the mucopolysaccharide? The mucopolysaccharide that absorb water in between. Mucopolysaccharide will absorb, they will absorb water. They absorb water in between, they will absorb water in between vitelline and plasma membrane. In between vitelline and plasma membrane, in between vitelline and plasma membrane, they will absorb water. So more amount of water will come in between, more amount of water, H2O, water will come between vitelline membrane and plasma membrane. In between vitelline membrane and plasma membrane, more amount of water will come. More amount of water will come. The third chemical that is present there, the third chemical that is known as peroxidase is present there. Peroxidase is present there. Peroxidase is present there, and transglutaminase, peroxidase and transglutaminase is present there. Peroxidase and transglutaminase is the next chemical that is coming from the cortical granule. What they will do, what they will do, they will do the cross-linking between the fertilization cone, very well, over this ova, fertilization cone is formed, right? Over the ova, this type of fertilization cone was formed when a sperm was coming. I hope you remember. What they will do, they will cross-link. They will cross-link peroxidase and transglutaminase, cross-link between. They will form cross-link between. They will form cross-link. In this manner, they will form cross-link between. They form cross-link. They will form cross-link. They will form cross-link between fertilization envelope. Fertilization envelope. They will form cross-link between the fertilization envelope. So we are discussing about the slow block. What is slow block? What is fast block? Fast block will happen within second. And what is slow block? Slow block is happening by taking time. It requires time. In order to prevent the entry of sperm, our body is doing slow block also. What is happening in slow block? A sperm is coming. When a sperm is coming, calcium channel will open. When a calcium channel opens, the cortical granule that is present in the ova, the cortical granule that will fuse with the plasma membrane, and they will release their chemicals outside. The released chemical contain cortical granule serine protease. What is the role of that cortical granule serine protease? They cleave the connection between plasma membrane and vitelline membrane. In between plasma membrane and vitelline membrane, a connection will be there. That connection will be removed. Second one, that contains mucopolysaccharide. What is the role of that chemical? That absorb water in between vitelline and plasma membrane. All the space will be now filled with water. Then what is there? Peroxidase and transglutaminase is coming. They form crosslink between the fertilization envelope. They form the crosslink in the surface of the ova. Another thing, the fourth chemical, the fourth chemical that is known as hyalin. The fourth chemical is known as hyalin. Hyalin also coming from the cortical granule. What is the role, They form a coating around they form a coating. They will form a coating. They form a coating around, they form the coating around the egg. They form the coating around the egg. So by doing all these things, what happens, no more sperms can enter into the ova after this. Only a single sperm that can enter into the ova. No further sperms can enter into the ova. Whether it is clear?
Yes, sir.
Okay. And the process is known as?
Mechanical blocks.
Okay. That is slow block, right? Slow block or permanent block. Okay. Ready. So, when a sperm reaches the egg, okay, sperm reaches the egg, egg gets activated. Whenever a sperm comes, the egg gets activated. Egg will be activated. We are discussing about those mechanisms. The first mechanism. So we are discussing about the mechanism by which the egg is getting activated. The first mechanism is known as, listen to me very well. Here it is a ova, okay? Here it is a ova. This is a ova membrane, okay? Here it is a ova membrane. Here it is a ova membrane. Here it is an ova membrane. There is a channel. Here it is a channel, okay? Here it is a channel. This is the channel. Whenever the sperm come, whenever the sperm, this channel is known as Na plus H plus pump, okay? This is Na plus H plus channel that is present on the ova membrane. So, whenever the sperm come into the ova, this is the ova's membrane, whenever a sperm comes into the ova, a compound from sperm. We don't know what is the name of that compound. A compound from sperm, what will do, a compound from sperm activates a tyrosine kinase. A tyrosine kinase will be activated by a compound from the sperm. That is tyrosine kinase, okay? Tyrosine kinase. What is the role of tyrosine kinase? Tyrosine kinase means that can phosphorylate, that can give a phosphate group to tyrosine residue. Tyrosine kinase means that can give tyrosine kinase. This is an enzyme that can phosphorylate. What they can do? They can do phosphorylation. Phosphorylation means they can do or they can give a phosphate. They can phosphorylate whom? Phosphorylate tyrosine residue into a tyrosine residue, into a tyrosine molecule. They can give a phosphate. That's only the meaning of tyrosine kinase. Okay. So the tyrosine kinase that is now present inside the ova, what the tyrosine kinase will do, the tyrosine kinase will activate, they will activate phospholipase C. They will activate phospholipase C. They can activate phospholipase C. So the phospholipase C, the PLC, phospholipase C will be cleaved into. Phospholipase C will cleave. The phospholipase C will cleave into. What is phospholipase C? Phospholipase C that will cleave into phosphatidyl inositol 4,5-bisphosphate that will cleave in this manner, PIP2. What is PIP2? PIP2 means phosphatidyl, phosphatidyl, phosphatidyl inositol, phosphatidyl inositol 4,5, phosphatidyl inositol 4,5 biphosphate, inositol triphos, biphosphate. That is the PIP2. Okay. So a sperm is coming, and sperm is activating a tyrosine kinase. That tyrosine kinase is activating whom? A phospholipase C. Phospholipase C cleaves into PIP2, phosphatidyl inositol 4,5 bisphosphate. So the phosphatidyl inositol 4,5 bisphosphate that will cleave into IP3. IP3 means inositol triphosphate. IP3 is inositol triphosphate. Inositol triphosphate. Inositol triphosphate. It will cleave into inositol triphosphate and DAG. It will be cleaved into inositol triphosphate and DAG. DAG is diacyl glycerol. It will cleave into DAG, diacyl glycerol. I hope you remember. I have clearly explained about all these when we were discussing about that glycerol and fatty acid and all. So IP3 is there and DAG, IP3 and DAG is activated. What is the role of the DAG? The role of the DAG is that what the DAG will do, DAG can activate Na plus H plus exchanger. DAG activate Na plus H plus exchanger. Na plus H plus exchanger was present in the egg, right? So they will be activated. So what will happen? The Na plus enters inside. Due to the presence of that, Na plus enters inside and H plus will move outside. H plus will be moved outside. So what will happen, So what will happen when it is activated, when it is activated, there will be high amount of Na plus, The level of Na plus will be high in cytoplasm, in ooplasm. Inside the ooplasm, more amount of Na plus will come. That will increase the alkalinity. When more amount of Na plus will come, more alkalinity will be increasing. More amount will be the alkalinity. When there is more amount of Na plus and more alkalinity, what will happen? That stimulate DNA synthesis. That stimulate DNA synthesis of the ova, DNA synthesis and RNA synthesis. DNA synthesis and RNA translation into protein. This process will happen. This process will occur inside the ova. In another manner, what is happening to the IP3? What is the role of IP3 now? IP3 is a calcium releaser. It is a calcium releaser, okay? IP3 is a calcium releaser. It is a calcium releaser. So IP3 will release calcium. IP3 will release calcium. IP3 will release calcium. IP3 is releasing calcium from endoplasmic reticulum. It will release calcium from endoplasmic reticulum to cytoplasm. Calcium from endoplasmic reticulum to cytoplasm. Whenever the cytoplasm contains calcium, then only the granule reaction. We have already said about the granule reaction. Then only the granule reaction is happening. This is the thing which is occurring in a ova after the sperm is entering. Once again, listen. A sperm is coming near the egg. When a sperm is coming near the egg, what will happen? When a sperm is coming near the egg, the egg gets activated. How the egg is getting activated? A particular chemical, a compound which is coming from the sperm. That compound, we don't know the name of that compound. That compound will activate one tyrosine kinase, TK. One tyrosine kinase is activated there. That tyrosine kinase, what is a tyrosine kinase? Tyrosine kinase is a chemical which can phosphorylate a tyrosine residue. This tyrosine kinase can activate phospholipase C. The phospholipase C, that can cleave. What is the role of the phospholipase C? That will cleave phosphatidyl inositol 4,5 bisphosphate. The phosphatidyl inositol 4,5 bisphosphate will be cleaved into IP3 and DAG. IP3 is inositol triphosphate, DAG is diacyl glycerol. What is the role of DAG then? DAG will activate the Na plus H plus exchanger. Na plus H plus exchanger will be activated. So more amount of Na plus will be enters into the ova. Whenever more amount of Na plus enters into the ova, the ova will become alkaline. Whenever the ova become alkaline, that stimulate DNA synthesis and protein production, RNA translation. The same manner, there is IP3. The IP3 is a calcium releaser. IP3 will do the release of calcium from endoplasmic reticulum to cytoplasm. From endoplasmic reticulum to cytoplasm, they will release calcium. What happened to them now? What happened to them? That will be doing granular reaction. They will do the granular reaction that we have already studied. That is the sperm and egg activation. Whether it is clear?
Yes, sir.
Okay. Now we have to discuss about the fusion of genetic material. Fusion of genetic material. How the genetic material is getting fused. Fusion of genetic material that we are discussing now. So who is entering the egg? Who is entering the egg? Sperm pronuclei. Sperm pronuclei. Sperm pronuclei means that is a nuclei of the sperm. And proximal centriole. Sperm pronuclei and proximal centriole. Sperm pronuclei and proximal centriole enters the egg. Who all are entering the egg? Only sperm pronuclei and proximal centriole enters the egg. They only enters the egg. What happened to the mitochondria? Mitochondria of sperm. Mitochondria is there in sperm. Mitochondria and tail was there, right? Mitochondria and tail, they will be degraded. They will be degraded. They are not at all required. They will be degraded. So here it is the ova. Okay, here it is the ova. This is the egg pronuclei. This is the, this is the ova. This is the ova. Okay, this is the ova. This is a pronuclei of ova. This is a pronuclei of ova. Here it is the egg pronuclei. This is egg pronuclei. This is a pronuclei of egg. Okay, this is a egg pronuclei. And who is entering inside? Who is entering inside? The first one, the sperm pronuclei will enter inside, right? Sperm pronuclei will enter inside. Sperm from sperm, its nuclei, sperm pronuclei is coming inside. Not only sperm pronuclei, in addition with who is also entering, the centriole is also entering. The centriole means that is a proximal centriole is also entering. This is a proximal centriole. So proximal centriole will be also entering. Proximal centriole and sperm pronuclei is entering inside the egg. What happens then, What happens then, After the sperm entry, now the sperm is inside, right? The sperm pronuclei, what happened to the sperm pronuclei, The sperm pronuclei will do decondensation. Decondensation will occur. Decondensation of pronuclei will occur. Decondensation of sperm pronuclei. I want an answer from you. The sperm nucleus or the sperm chromosome is highly condensed with the help of a particular type of histone. What is the name of that histone? Tell me. A particular type of histone. A particular type of histone is there. With the help of that histone, it is highly condensed. The sperm pronuclei is highly condensed. What is the name of that histone? Anyone? Come on.
H1.
Huh?
H1.
No. Huh? No. We only took the class 2 days back. The spermatid is getting converted into spermatozoa. Whenever the spermatid is getting converted into spermatozoa, a special type of histone is used there. That histone is known as, I am asking the question, that histone is known as, I think you are simply sitting in the class. You are just making notes. And after that, you are just keeping your notes aside. You are not making, you are not having time to study. You are not making an effort to study, I think. If it is a developmental biology class is going on, then you have to study the DB first, right? Without studying the previous class, how you can understand this class? Don't sit as like you are watching a movie. You are taking efforts only for you. So you have to study. Sperm pronuclei or sperm nuclei is condensed with the help of a special histone. That is a histone variant. I have told everything. Just look back, go to YouTube channel and look the video. There is there. You can expect it. That's all. All those things are sure mark questions. What you are thinking? You are simply thinking, okay, anyhow, already joined. Okay, anyhow, we can complete the classes. It is not like that. We are taking the effort only for you. I can simply log off now and I can go. What will happen? Who will be asking me? Who will be responsible then? Why we are spending why we are spending this time, you know very well what is the minimal amount that we are collecting and all, everything, we are in a motto to spread the awareness regarding life science. That's why we are taking all the effort. So it is your responsibility. You are our gems. We only have you with us. In yours batch at the beginning, 19 members were there. Now you are only 7 members. Now you are only 7 members. Even if it is only one student is left, we will take the class. But how to study? That is your responsibility. We can only teach. Study is from your side. When I am asking a question, when I was asked this question in last class also, you all were keeping mum. It is not. It is not at all okay. When I am asking something, okay, I want the prompt reply. Spontaneously, I need the answer for that. Then only I will be happy. Okay, inside the sperm, inside the sperm, there is a special type of histone. That histone is known as protamine. That histone is known as protamine. I hope you may be hearing this first time. Just look into the previous class video. It will be available in YouTube. Just check over that. Protamine histone is required for sperm condensing. Anyhow, what will happen, once the sperm is entered inside the ova, once the sperm is entered into the ova, what will happen, that protamine will be replaced by the protamine will be replaced by normal histones. Protamine will be replaced by normal histones in ooplasm. Normal histone in ooplasm. From ooplasm, the protamine will be replaced by normal histone. And after that, what will happen, our proximal centriole, the proximal centriole, he will do a 180-degree rotation. The proximal centriole will do 180-degree rotation. The proximal centriole, what they will do? 180-degree rotation. They will do a 180-degree rotation. Sorry, there will be a 180-degree rotation from 180-degree rotation from, 180-degree rotation will be coming from the sperm pronuclei. The sperm pronuclei, the sperm pronuclei, he will do a 180-degree rotation. Sperm pronuclei will do 180-degree rotation. Whenever they are doing a 180-degree rotation, what will happen now, what will happen? There is two, this is a sperm pronuclei and this is a ovum pronuclei. The proximal centriole will come in between. The proximal centriole will come in between them. Then what is the role of the proximal centriole now, The proximal centriole act as microtubule polymerizing center. It will act as a microtubule polymerizing center. Who will act? The Proximal center, this one will act as a microtubule organizing center. The proximal centriole will act as a microtubule organizing center. Then what will happen, from the microtubule, from the centriole, what will happen, the microtubule will come and the microtubule will attach on the microtubule attaching on, microtubule will come and the microtubule will be attaching on both the nucleus. So whenever it is attaching with both the nucleus, it will pull it towards, the nucleus will be pulled towards the centriole, nucleus will be pulled towards the centriole. As a result, the two nuclei will be merged. The two nuclei will be merged and produce a diploid nucleus. It will become as a zygote now. Then it become as a zygote. Zygote contains 2n condition of nucleus. Now it is known as a zygote. It is known as zygote. In this manner only, the sperm and ova fuses inside the ova. Sperm nuclei and the ova nuclei fuses inside the ova. So we are looking for the fusion of genetic material, how the genetic material is getting fused. Sperm pronuclei and proximal centriole only, they will be entering into the egg. To the egg, okay, sperm pronuclei and proximal centriole will be only entering into the egg. Then the mitochondria, the mitochondria and tail of the sperm, what will happen to them? They will be degenerating. Whenever the sperm pronuclei and the proximal centriole enters into the ova, whenever they enters into the ova, what will happen next? The sperm pronuclei, decondensation of sperm pronuclei will occur because it is highly condensed with the help of protamine histones. So the protamine histones will be replaced. The protamine histones will be replaced by normal histones in ooplasm. So after that, the sperm pronuclei, they will do a 180-degree rotation. With the help of that 180-degree rotation, the sperm pronuclei will come in between. Now the sperm pronuclei will come one end and the ovum pronuclei will come in another end. And in between, there will be the centriole. The centriole will be in between both. Then from the centriole, what will happen? Centriole will act as a microtubule organizing center. Centriole will act as a microtubule organizing center. Microtubules will come from centriole. Microtubules will go towards both the end and they will pull the nucleus together. Whenever they are pulling the nucleus together, they will become as a diploid zygote. It is a formation of zygote. Now, a zygote is formed. Whether it is clear?
So,
When I am asking, you are okay. Just 1 second. I am clearing this. Just